Hello and welcome, Matric learners for life sciences. So we are welcome here today with Aslam. Aslam, how are you doing?
I'm doing great. What you say today? What's happening?
Yeah, I'm all good, guys. Guys, remember, everything we teach you today in the lesson will be on our Facebook page. That's facebook.com/learn extra. On our page, you'll be finding the instructions to win one of these. All you have to do is answer the test question, the test yourself question, and you could be walking away with a brand new Casio calculator, sponsored by Casio, of course. But guys, let's get on to the lesson, and I hope you guys enjoy. Aslam?
Thank you so much, guys. Guys, welcome back. I'm hoping you've enjoyed your holidays and you didn't leave your books totally marooned on an island. You looked at them from time to time and you kept in touch with your work. It's very important in this year, especially grade 12s. I was telling the matrics today again that if you look at this term, there's only 6 weeks left in this term. And if you equate that to school days, it's about 25, 26 days only. And then you start your trial exams. And guys, from then on, it's just exams. You come back after your holiday and exam starts again. So you don't have time to mess about now. Keep in touch with your work. The other thing is, as you can see on the board, we are revising the June exam questions. We've picked from 2 provinces for now, and that is Gauteng and KZN. The reason behind this is we need to change our mindset in terms of exams and assessment. Many of you think that exams is there just to give you a mark that goes onto your report so that you can look good or you can look bad if you didn't do too well. That is not the purpose. The purpose of an assessment is to evaluate the teaching that has gone on, whether you have understood the concepts. Remember, these concepts that you are learning may not only be for now, that this exam, let me write this and get an A, or let me get a B or whatever and move on. You may choose a course next year in varsity where the foundations that you've learned in math, science, accounting, life sciences, in any of the subjects, you may need to take that foundation and build on it next year. And if you do not have this foundation, you're going to be the loser. So please make sure that you are concentrating when we're doing the work. And also, guys, the other reason is you haven't scored full marks in your paper. You need to know where you have gone wrong. And if you're from another province, you're going to see the other papers as well. And if you are in another province and you can send us your papers, by all means, send it to us via the Facebook page so that we can use your questions as well. And send your questions, your individual questions if you want to today as well, and we can address them as well. The other thing, guys, if you look on the Facebook page, there is a post on a water challenge that is being held in this country. And remember, apart from answering those questions for the challenge and whatever prize there is, human impact is in your syllabus. It's plus minus 40% of paper 2, or paper 1, I should say. And when you answer these questions, it's going to sharpen your skills in terms of water. And remember, under human impact, there's a section on water security, water pollution, etc. So this will tie in with that as well. So be sure to check the link out and enter that competition as well. Let's go to our questions now. What do we want to do? We want to revise questions appearing in your June exams. But remember, they may not have been called paper 1 and paper 2 in your particular province. Most provinces would have written one paper that had questions from paper 1 and paper 2. What we have done, we have extracted those questions that are relevant to paper 1. Why have we done this? So that you also know what is paper 1 and what is paper 2. Remember, it is different from 2013. Your best guide at the moment is the exemplar paper 2014, paper 1 and paper 2. Those will clarify which topics are in paper 1 and paper 2. Not that we haven't done it here already. We've also done it in the winter school. We've given you the whole breakdown for paper 1 and paper 2. So we want to cover that, the work that is covered in terms 1 and 2. first question, or the challenge question rather, I must get used to the whole setup again. I'm getting rusty. I want to rush into the question paper. The challenge question, as usual, we want to challenge you first. A very simple sounding question, but you need to put your thinking caps on to answer it. So the broad stem of the question says, predict what would happen if. So in two instances, you need to do a prediction. What is that prediction? A, the ciliary muscles of the eye lost its ability to contract. The ciliary muscles of the eye lost its ability to contract. And B, the circular muscles of the eye lost its ability to contract. I'm only talking about contract here. Yes, that's what I'm talking about. But note, here we are talking about ciliary muscles. And here we are talking about circular muscles. And the reason I put that question there is many of you confuse the two, ciliary muscles and circular muscles. So obviously you need to look at the diagram of the eye first again. Where are the ciliary muscles? Where are the circular muscles? And work your way from their functions to answer that question. I will leave you with that. Please post your answers on the page so that we can follow up as we go along. Right. All right. Let's go to the real questions now. The first one, adapted from Gauteng, June 2014, question 3.3. As you can see from the diagram, or before we even go there, the diagram below shows a cross-section through the ovary of a human reproductive system. So it already tells you that we're dealing with reproduction. And guys, this is something you need to also get into your head, that when you look at a question, the stem will already tell you either which chapter it is, or the diagram will be a clue. So you need to put yourself into context. Okay, right, this is about reproduction. This is my body of knowledge in reproduction. Let me use that knowledge to answer the question. So let's look at that now. The question says, answer the questions based on the diagram. They're giving you a diagram there of the ovary and three labels, A, B, and C. And the first question asks you to label A, B, and C. And obviously, the first one, A, is the Graafian follicle. The Graafian follicle. B would be the ovum. And C is the remainder of this Graafian follicle after the egg has been released, and that would be the corpus luteum. By the way, the question only asked for A and C. Aslam already gave one extra answer, but that was good because it's going to come back later to bite us anyway. But remember, follow instructions. This question asks you for A and C, not A, B, and C, not A to C, but A and C. So you would have answered it accordingly, only A and C. Identify structure label B. We were forward, we were precocious, and we have answered that already, and that is obviously the egg or ovum. Right, the right terminology, ovum. Give the ploidy. Ploidy. Now, in your studies, where did you hear about ploidy? This word ploidy, where does it come from in your studies in grade 12? There's only two words that you have learned with this ploid in it, and that would be haploid or diploid. So, is this structure B haploid or diploid? This is what the question is asking. And obviously, because it's a gamete, it would be haploid or N. Give the name of the hormone secreted by structure A. Structure A was the Graafian follicle. So the hormone that is only one hormone. Notice it said the hormone. It's not hormones, one hormone, and that would be estrogen. Hormone, estrogen. Provide one function of the hormone identified in question 1.3a. What is the function of estrogen? Firstly, in the females, the first function would be to bring about secondary sexual characteristics in the female. Okay. Secondary, female characteristics, and more importantly, well, I can't say more importantly because they're both important, but in terms of our study of reproduction, this is core. It stimulates the thickening of the endometrium. In other words, it causes the uterine lining to become thicker. Why? For possible pregnancy, for possible implantation. And sometimes it also not sometimes, it also stimulates the production of LH. When estrogen is high, LH will be secreted after that. Discuss how oogenesis is prevented once a female is already pregnant. Oogenesis prevented when a woman is pregnant. Which hormone is high in concentration when a woman is pregnant? FSH, estrogen, progesterone, or LH? You got it? Progesterone is the one that's high. And what do we know about progesterone? A high level of progesterone inhibits FSH. In other words, when the levels of progesterone are high, FSH will not be secreted. Now, let's think for a moment. What is FSH? Follicle stimulating hormone. So if the follicle stimulating hormone is inhibited, it will not stimulate the follicle. Good, you got that. And if it doesn't stimulate the follicle, then oogenesis cannot take place. Because where does oogenesis take place? In the follicle. Good. So let's look at that answer again. When she's pregnant, the progesterone levels will remain high. Key is not about that there is progesterone, but the progesterone is high. You must mention that. Those two must be tied together. This high progesterone inhibits the secretion of FSH. And it will not stimulate the formation of ova or the follicles, and therefore no oogenesis will take place. So in other words, this was another way of asking you questions on reproductive without just telling you to write an essay on it. My finger seems to be working better than a pen today. Okay. Now, also from Gauteng, records of human fertility for the period 1941 to 1990 have shown changes. We need to worry about this one. Have shown changes in the sperm counts of normal men. The table below summarizes the changing percentages of men with high or low sperm counts over a period of 50 years. The changing percentages. So there's a table, 1941 to 50 and so on. And they're telling you here in the first column here, men with high sperm counts, the percentage was 50, then 45, 28, 21, 15. You can see what's happening there already. And on this side, men with low sperm counts, 4, 4, 5, 11, 14, 18 in 1990. And we are now in 2014, so we need to start worrying. On the same system of axes, draw two sets of bar graphs to compare the percentages, not the sperm count, but the percentages of men with high sperm counts with those with a low sperm count over the 50-year period, 1941 to 1990. And we're not going to waste too much time. Just going to open that up. So the heading, the title, must have both variables. And the variables are, one, the percentage of men with high and low sperm counts, and the period from 1941 to 1990. So over time. Okay. The x-axis heading, time period in years. Notice the unit must be given. And here, because you're using the word percentage here, then that's the unit on its own already. Percentage of men. Okay. If you want to, you can put that in brackets there, percentage, but it's given there already. Notice there's a key also. The one that is shaded, high sperm count. The one that is unshaded, low sperm count. So there's a mark for that as well. Okay. Notice the intervals, 10, 20, 30, 40, 50. They are equidistant apart. And the bars, the width of the bars are the same. and the distance from one set of bars to the other set of bars is also the same. These are all graphing skills that you need to have. Obviously, the plotting is easy. You need to just plot them according to what the table gave you, and you will then get your marks for that accordingly. There were 7 marks given there. All right, the 1 mark would have been given there, 1 for this whole idea here and the interval, 1 for this whole idea here and the interval or the bar's width. That is 1, 2, 3, 4, and you would have got 3 marks sometimes for, oh no, 1 mark for the type of graph, and 2 marks for plotting sometimes, and that will give you 7 marks. Good? Describe the trend for men with low sperm counts and compare it to those with high sperm counts. The trend that you saw, and I highlighted that to you on the table. You should have picked that up already. Without the graph, you would have picked it up with the table already. As time progresses, we're comparing it with time. There's 2 variables. One is time and one is the percentage of men. So that's what you have to do. And generally, when you're describing a trend, you must start with the independent variable. You can't say as the percentage of men increase, so too does the time. Doesn't make sense, because the number of men is not affecting time. Time is affecting men. Okay? So as the time progresses, the percentage of men with low sperm counts increases. It doesn't say that the sperm count decreases. That's not what it's saying. It's saying that the percentage of men that have low sperm counts, it increases. And the percentage of men with high sperm counts decreases. Lots of learners, and this one here would have been a ridiculous one to start off with the number of men. But in other examples, lots of learners, they switch the data around. So they say, for example, let's say enzyme activity and temperature. We know that when an increase in temperature occurs, the enzyme will become denatured, so therefore its activity will decrease. Now what learners do, they say as enzyme activity decreases, the temperature increases. Now that can't be right, because the dependent variable is dependent on the independent variable and not vice versa. So in other words, the enzyme activity is the dependent variable. It depends on temperature. Temperature does not depend on the enzyme activity. Hence, it's called the independent variable. So that's where we have this answer here. Lots of learners confuse it just because of the independent and dependent. Remember, whenever they ask you the relationship between 2 variables, start with the independent variable, because it is that variable which affects the other variable. With that,
so guys, it's turning out to be a very interesting lesson. Stay tuned. We're just going to go for a short ad break. Remember, all this can be found on our Facebook page, as well as the competition for this Casio calculator, but we'll be back in a few minutes. Stay tuned. Hello and welcome back, grade 12 matric learners for life sciences. Today we're here with Aslam, and he's going through the June paper for Gauteng. But if you're not in this province, you can still follow along. Take notes. All the notes can be found on our Facebook page, facebook.com/learnextra, and you can download those papers as well as any work we do today. There's also a competition for this Casio calculator. Remember, you can enter that as well on our Facebook page. And don't forget about the water game, watergame.co.za. That can lead to you winning 5,000 Rand as first prize. So get over there, play the game, and you never know, it could be you. So without further ado, Aslam.
wow, 5,000 Rand.
5,000 Rand for
Plus
first prize.
you're learning human impact. What a bonus.
What a bonus
5,000 Rands plus 50 marks.
plus 50 marks.
Okay, you may get quite a bit of marks because you're learning something about human impact in it. It may be appearing later, you never know, so you need to look at that. Okay, study the graph. We're still busy with reproduction. I've kept them in sections so that we do one section and we've covered both sides of the spectrum now. The next one is if they give you a graph, and this one was done in KZN, question 2.3. Study the graphs below showing the levels of the hormones involved in the menstrual cycle in most women. So there we go. We have this graph, famous graph that we normally see, more or less, not always exactly the same, but at the bottom, we have the days there, and in this block, we have the ovarian hormone levels, and in this block, we have the pituitary gland hormone levels. So we're getting two sets of hormones. You need to know which hormones are secreted where, and I'm just going to put here our famous word, FOB, and those are the hormones that you need to concentrate on, but you need to know which ones are secreted by the pituitary gland and which by the ovary, in the ovary, okay? So they're giving you the story there. We're seeing what's happening there. There's the hormone A, B, C, and D. And there's your day 14. There's the beginning of the cycle, middle of the cycle, and end of the cycle. So let's get into the questions. Provide the label for hormone A. Now, to identify hormone A, you have to look at what I told you here. What did we write there? FOB. And if you remember, FSH, estrogen, LH, and progesterone. Why do we write it as FOB and not FLOP? Because that is the order in which these hormones increase in a menstrual cycle. So we look at the one that increases first in the cycle. We look at the one that increases first in the cycle, and that would be this one here, that's high, that's B. And then this one here, A, peaks in the middle of the cycle. You're looking at it where it's increasing. After B increases, you see there's an increase in C, and last, there's an increase in D. So based on where we found the increases, we can place them. That B will be FSH, C will be estrogen, L will be LH, that's A here, because it peaks in the middle, 14th day, and the one that peaks later, D, would be progesterone. So once you've identified that, that is why this hint and clue is always important for you. So you write that down and you identify it already. You can write it on the graph or whatever. So they're asking for hormone A, and already we know from our identification that that's LH, luteinizing hormone. According to the graph, during which period of time is the level of hormone C lower than that of hormone D? When is hormone C lower than D? If we look at this whole period from 0 to 14, C is higher. You do agree, I'm hoping? All of that period there, C is above D. Till here, it is still above D up to that point. And from here onwards, we find that it is below up to the 28th day. So it's somewhere in between 14th and 21 days there and the 28th day. So that's the period that we are looking at. Again, you didn't need to be a rocket scientist to figure that out. You had to read from the graph. From the 16th or 17th day to the 28th or 29th day. Name and explain the relationship that exists between the hormones labeled B and D in the menstrual cycle. And I'm not going to waste a lot of time discussing this. I'll tell you why just now. B, FSH, and D, progesterone. The relationship. This is the relationship we just spoke about in the last question. Same thing. Notice the question is different, but the answer is the same. First of all, they ask you to explain the relationship. This relationship is called a negative feedback mechanism. You needed to say that first. Thereafter, you say increased levels of progesterone will inhibit the hormone B, and therefore it's going to decrease the number of follicles. Or you can say then when the levels of D progesterone decrease, the B stimulating hormone will be released from the pituitary gland because it's a negative feedback mechanism. So you're explaining the relationship. Okay? Describe the changes that occur in the ovary during the 28-day cycle. Now, without this graph, you could have answered this question because it's just telling you to tell us what happens in the ovary. In the 28-day cycle. But you had a graph to help you. In the 28-day cycles, follicles are released. Follicle grows and matures into the Graafian follicle. On approximately the 14th day, the ovum is released. All right, they're writing it there. Ovulation takes place. The remains of the Graafian follicle becomes the corpus luteum. Description, you're just describing the events that are taking place under the influence of LH. The corpus luteum, you can even say that secretes progesterone. You can add that, but remember, you're not going to talk about the action of progesterone because they did not ask you what's happening in the uterus. Okay? And the corpus luteum secretes progesterone. If fertilization does not occur, it degenerates quickly. That's about it in a 28-day cycle, especially in this 28-day cycle. What is happening to progesterone in the end? Can you see what's happening? It is decreasing. If progesterone is decreasing by the 28th day, it would mean that this woman is not pregnant. That is why, because corpus luteum has degenerated, progesterone levels have decreased, and you can see what's happening to estrogen there later. It will start increasing as well. And look at FSH there. FSH first. There. FSH starts increasing, and then estrogen will start increasing thereafter. Okay? So you need to be able to see these clues on a graph, and you'll be able to determine whether a woman is pregnant or not. Good. Come on. Hey.
Bit of a love-hate relationship with the white.
Back and forth. Okay, we still with the same question. If fertilization occurs on day 15, so in other words, after the egg is released and fertilization takes place, describe the changes that occurred in the fertilized egg until the embryo attaches to the endometrium. Again, a description of a question. You're only going to describe what's happening to the fertilized egg. First of all, the zygote will divide by mitosis to form a solid ball of cells called the morula, the many-celled stage. The morula, further division takes place, can become the blastocyst and blastula. There's much more to it. There's more intricate details in this process, but for your exam, this is the detail that you need to go. This is the depth at which you need to go. The blastocyst or blastula develops into the embryo, which then attaches onto the endometrium or becomes implanted onto the endometrium. That is what, up to that point, they needed you to explain. A woman takes a temperature reading on a daily basis during her menstrual cycle. The table below shows her temperature records for days 11 to 19 of one of her menstrual cycles. A slight drop in temperature followed by a sharp rise in temperature indicates that ovulation has occurred. A slight drop in temperature followed by a sharp increase, rise in temperature will tell you that. So day 11, 36.5, 36.6, 36.6, 36.6, 36.6, 36.2, 37, 36.9, 36.9. So you need to look at that. According to the graphs, on which day do most women ovulate? According to, not the graphs, but the table in this case. It would be day 14, around there. According to the graph, sorry, we could go back to the graph there. The other one there that said the graph would say that most would be on 14. And according, some women use the information in the table to prevent themselves from falling pregnant, use the information to explain why this method of pregnancy is not reliable. Now look at this table. We said that most women ovulate on day 14. Here, what do we notice? When does the temperature drop? It drops on day 16 and rises on day 17. So this woman's ovulation probably took place there. Okay? So let's see what we have to offer there. The woman ovulated on day 16 or 17. We could have seen that there. Indicating that the days of ovulation can vary from woman to woman. And in the same woman also. 1 month it may be on the 14th, the next month it may be on the 15th, and so on. There's so many other factors that can happen. Stress can cause it to be the ovulation to take place quicker and so on. And if they avoided intercourse on day 14, she could still have fallen pregnant after that because ovulation took place after that. And anyway, just remember, the sperm can last in the woman's body for up to 72 hours. The sperm can remain alive for up to 72 hours. So the chances of playing with that, if you do not want to fall pregnant, not a good choice. Okay, let's move on to see something now. And we go to Gauteng, June 314. Diagrams 1 and 2 show a section through a portion of a human eye. Study the diagrams and answer the questions that follow. Good. There's a clear diagram given to you there. They've labeled two things, A and B. Which diagram represents the eye when a person is reading a book? Now, we have to ask ourselves, which part of the eye is involved in focusing? And reading a book would represent near vision. And your answer would be the lens. What must happen to the lens when we want to read a book? The lens must be more round or more convex. Okay, so let's look at the diagram. It's obvious from the diagram then that the lens on the right, diagram 2, is rounder than the one in diagram 1. There is no A, there is no B in terms of the diagram. There's labels, yes. But the question asks which diagram. And if you give the answer A, your answer is incorrect because there's no diagram A here. There's a label A. Your answer had to be diagram 2. Okay? Name and describe the process that causes the change from diagram 1 to 2. In other words, how do we change the eye to see near? And that's known as accommodation. First, we have to name it. Accommodation. Prefer the yellow. Accommodation. So, here the ciliary muscles contract, causing the ciliary bodies to move towards each other. They contract towards the lens. And when we were teaching the section, I stressed that point, that the ciliary muscles contract towards the lens. And the tension on the suspensory ligaments decreases, causing the suspensory, I want to add one part there, causing the suspensory ligaments to become slack. That means they're not tight. And if they are slack, there's less tension on the lens. And the lens then becomes more convex. Let's look at a diagram. Here's the suspensory ligaments here. Okay? Your ciliary body muscle is located here where this circle is. So if it contracts towards the lens, it's not pulling the suspensory ligaments. And the ligaments are not tight, they're not pulling on the lens. So the lens takes a round shape. Whereas in this diagram, as you can see clearly, mind the pun on see clearly and the eye in one sentence, there you got your suspensory ligaments that are taut. The word is taut, T-A-U-T. That's because they, look at the size of that circle now, ciliary muscles. They have pulled away from the lens. So they contracted away from the lens. And when they contract away from the lens, they're pulling the suspensory ligaments tight or taut. And when the suspensory ligaments are taut or tight, they're pulling the lens, you know, China eyes, you want to call it. Just now somebody says, I am what? Homophobic. All right, let's move on. Discuss how structure B is suited to its function. What is structure B? It's the outermost layer of the eye, the white of the eye, and this is known as the sclera. When they ask you suitability, you must first know what we're talking about. The sclera's function, we must also know, is to protect the eye and to maintain the shape of the eye. And to do this, what does it have? The sclera is tough and non-elastic. Oopsie. Sclera is tough and non-elastic and therefore helps to protect and to maintain the shape of the eyeball and provide protection there, right? An unequal curvature of structure A can cause visual problems. Unequal, the key is unequal. Structure A, again, let's go back to structure A, is the cornea, that part, the cornea. So they're saying unequal curvature. And the word unequal is the clue here. That means the person is suffering from, you guessed, astigmatism. That's correct. How can we treat it? We use glasses or contact lenses or laser strategies. And when we're using these glasses or lenses, these glasses or lenses must be unequally ground. They must also be unequal to compensate for those parts that are lacking on the cornea of that person. That's what we have to do there. Good? All right, another question on the eye. Provide labels for A, B, and D. Again, we've discussed this already, so it's going to be easy. Ciliary muscles, A. All right. Suspensory ligaments, B. And D is the cornea. We discussed that just now in the last diagram. Write down the letter and name of the part that provides color to the eye. That would be the iris. And the letter would be C in this diagram here. Iris. Provide the term used to describe the shape of part labeled E. Labeled E is the lens. In this particular diagram, that would be more convex, biconvex. Which diagram 1 or 2 represents the eye of a person looking at an object that is closer? That's easy again. It would be diagram 1 in this case. Notice the questions are similar. Two different examiners. They haven't spoken to each other, but similar questions coming out. This is the need for going over the papers, because this is the way that you have to learn for your work. Is the refractive power of the lens smaller in diagram 1 or 2? Refractive power, is it smaller in diagram 1 or 2? Obviously, the rounder the lens, the more its refractive power. So your answer is diagram 2. A rounder lens has a greater refractive power. And with that, we go for the break.
Cool, Aslam. So guys, I hope you guys have been finding this very interesting. Remember, you can still win this Casio calculator. All you have to do is go over onto the Facebook page and follow the Curio link on that page. Type in the code, answer yourself your test yourself questions, and you could be walking away with this brand new Casio calculator. So guys, we'll catch you back after the break. Welcome back, Matric learners, to this life sciences lesson on a Wednesday afternoon. I hope you guys have been enjoying the lessons so far. Guys, so just so we're clear, there's another way to get your hands on some mindset material. All you have to do is go over to intel.co.za, register yourself on Intel's site, and you can get your hands on some amazing mindset material. And guys, remember, we still have this Casio calculator up for grabs. There's also the water game. Remember, go over to watergame.co.za and you can win yourself 5,000 Rand. All you have to do is play the game. So, Aslam, I have a question here from one of the learners. Sibusiso Lawrence. Lawrence has asked, how do we draw a graph when we're not given the percentages? How do we calculate that?
Okay, the question that we were given, a graph, the percentage were given. Look at the table. Men with high sperm counts and men with high count, the percentage was given. Good. So in this particular case, the percentages were given to you, right? Now, Sibusiso wants to know, what if, I'm assuming this is what he wants to know, or because I'm assuming he did see that this one had percentages. What if they didn't give you percentages and they gave you numbers? That these were 50 men and 45 men and 28 men and 21 men and 15 men. All you do then is you total this column, you get your total, and each of these you work out a percentage, 50 divided by the total times 100 over 1. And you work out the percentage for individuals. And the same for that one. Remember, there are two separate sets of data, so you can't add all and then say that 50 out of this total, because these were dealing with men with low sperm counts and these were dealing with men with high sperm counts. And that is how you do it. So it's a simple scale. I'm glad you asked that question so that others know that if you are faced with that, what do I do? How do I convert it to a percentage? I'm hoping you got that one. Good. Let's go to the challenge question now. Did anybody give us some answers there, by the way? Challenge.
I haven't come across any yet.
Okay.
No.
Guys, you are slow on the uptake today. Nonetheless, I suppose winter blues and getting back to school blues, you still getting with the program. You should be back with us again next week.
Oh, we do have actually a few answers. Sorry,
give it to us. Let's see.
Well, it was about the eye. So the questions were about
the ciliary muscle, first one.
Okay, well, Sorry, I'm having a bit of an issue.
No, that's
Okay,
fine.
well, there we go. Sibusiso also commented, but let's see what you have to say, and then I'll come back to you on what
All
they
right.
say.
Okay, right. While we're getting that right, let's see what the answer is. It's very simple. If we looked at the last question gave us the answer to this. Because we said that for near vision, the ciliary muscles have to contract. Okay, so if the ciliary muscle remains, if it can't contract, that means it's remaining relaxed. And if it remains relaxed, it's moving away from the eye, all right, from the lens, sorry. And this will cause the suspensory ligaments to be taut, T-A-U-T. Thus, the eye will be fixed for distant vision. Why? Because, in fact, I can add there, the lens will remain flatter and the eye will be fixed for distant vision, so the person would need lenses, spectacles or surgery to facilitate near vision. And do we have anything close to that?
Yeah. Siabulela came in with the answer for B now.
Right.
And B's answer, he said, was the radial muscles will be unable to relax. Therefore, the pupil will be unable to constrict. Therefore, the amount of light entering the eye will not be controlled. Is that correct?
Good, he's on the line. The main thing in his answer is that he's located that the circular muscles got to do with the iris and the pupil. But your answer is general. Here the question was specific. If the circular muscles of the eye lost its ability to contract, you have all the elements of the answer. You would have probably got a few marks out of the total if you gave this in an exam. But remember, this question said if the circular muscles would not contract. Now, what happens then? In that case, if it doesn't contract, again, same thing, it will remain relaxed. And if the circular muscles are relaxed, the pupil would be dilated. Like he said, it would not be able to constrict. Right? So it's the same. Remember in the exam, we would give you that mark. That's 1 mark there. And then you said that the ability to control light would be lost. You must be more specific. You must say that this could affect vision because in brighter conditions, because too much light will be coming into the eye. You must specify because it is in that situation contracting that the pupil will be too large and too much light is coming in. To get full marks, to get the full benefit, that separates your average from the brighter, because you want mine brighter and brighter in this particular, what you call it, but you know what I'm saying. So if this one said relax, then your answer would say contract here, the pupil would be constricted, and in dim light the person would have a problem because there'll be too little light coming in. You need to be specific. But can you see the difference? I'm hoping my aim is met, that you know now that the circular muscles are associated with the dim and bright and pupil and iris. And the ciliary muscles are involved with near and far and the lens. This is what I wanted you to hone in there with that. Good. Okay, we're going to start having war here again. All right. We move towards the essay questions in both papers, and for some strange reason, both essays came from paper 1 items. The safety of an organism is ensured by its ability to receive information from the environment and then to respond adequately and effectively to that stimulus. Tyra is sitting quietly on a park bench when she hears a loud growling sound coming from behind her. Describe the events that result in her sound being interpreted by her brain and the effect that the fear will have on the sympathetic nervous system. Many learners would take the sympathetic nervous system for granted and they won't learn it. So when it was asked here, normally people will ask about adrenaline here. Here the examiner asked for sympathetic nervous system. And if you know your work, you'll know that the functions of sympathetic nervous system and adrenaline are similar. So there's 2 things that we need to do here. Notice this is information given. Now, here's the thing. Describe. There's the verb. After the verb, what are you asking you to do? Describe the events that result in sound being interpreted by her brain. And that is equal to hearing. And the effect that the fear will have on the sympathetic nervous system. That is equal to the functions of the sympathetic nervous system. These are the two things you needed to put in this essay. And let's look at it quickly. The first one is interpretation of sound. The growling sound causes a vibration, which is captured by the pinna. That's where we're starting. Captured by the pinna. The sound wave is captured by the pinna and directed towards the auditory canal. From there, it causes the tympanic membrane to vibrate, which in turn causes vibration in the ear ossicles. You can name them hammer, anvil, stirrup. This then causes the oval window to vibrate. So what are you doing? You're just following the vibrations with the structure of the ear. This is what you're doing in the first part. It has to be in a logical sequence, which in turn causes pressure waves to be set up in the cochlea. This is the inner ear now. The organ of Corti then converts the stimulus into an impulse, which then goes to the auditory nerve, to the cerebrum, where it is interpreted as sound. Sympathetic nervous system prepares the body for emergency, just like adrenaline. In fact, if somebody is in short supply of adrenaline, the sympathetic nervous system takes over that function. Good? So that's the first thing. It is mostly stimulating in effect. It stimulates the body into action, whereas the parasympathetic nervous system takes the body back into a relaxed mode. What does it do? It causes an increase in the heart rate and the production of adrenaline, and release. Production and release of glucose and or the conversion of glycogen to glucose. Let's look at that. Increasing heart rate, why? Increased blood flow, increased oxygen to the cells, removal of carbon dioxide. At the same time, we need energy, so therefore we need more energy. So these two things, both of them here, they both increase glucose. All right? Furthermore, causes the dilation of the pupils. Why? So that more light can enter so you can see the situation. Dilation of the bronchioles. These are the tubules in the lungs. Why? So airflow improves. So you can take in oxygen faster and release carbon dioxide faster. It inhibits peristalsis. You don't need to digest food when you're scared. That's not an essential function of the body at that time. You need energy, you need to think, and you need to move. It inhibits the contraction of the bladder. So you're not going to empty out your bladder at that time. Although some people may, in a fear situation, disagree to what I'm saying. It decreases the secretion of saliva and stimulates the secretion of adrenaline. So adrenaline does come in as well. All of these effects prepare the body for emergency situation. Some would also talk about sweating and other functions of the sympathetic nervous system. You could have included that in your answer. And here's your mark given. Only relevant information to hearing and sympathetic nervous system. If you spoke about something else, seeing, smelling, and all the rest of it, or any other thing, you would have lost mark. Or balance in the ear, you would have lost the mark. The events described for hearing, especially, are in the correct sequence. The sympathetic nervous system is logical because it's only the functions. And for comprehensiveness, all the structures that vibration pass through. That means you have to say everything in the ear. And at least 6 effects on the sympathetic nervous system. This is how you get your RLC marks. The relevant, the logical, and the comprehensive. They're not together. Each one is on its own. You can lose one and get the other 2, or you can lose 2 and get the other 1. It all depends on how you have answered this essay. And in KZN, they said, describe the mechanism by which a tightrope walker is able to balance using his ear. They want only how the ear is helping in this. And again, here it's simple. You're talking about the cristae in the ampulla at the base of the semicircular canals, stimulated by the changes in direction and speed of movement. Which way is he moving? How fast is he moving? The maculae in the utriculus and sacculus are stimulated by changes in the position of the head in relation to the body. So these 2 things are informing him. Am I moving forward? Am I moving backwards? Is my head upright? Etc. And with these things, these stimuli are converted into impulses which go via the, not the auditory nerve, but the equilibrium nerve. Okay? The equilibrium nerve to the cerebellum. Notice The other one, when we were hearing, that goes to the cerebrum. Now this one goes to the cerebellum because the function of cerebrum, balance and coordination. The cerebellum sends appropriate impulses via motor neurons to the muscles of the body to restore. Which way must I put more pressure so that I can be balanced on this tightrope? That's what we are saying. Don't go into too much details. Whatever is here, that's all you need to know. There is more details. There are more details, but you don't need to know all of them. And the essay here was explain how a person hears the sound of a dog growling and is able, notice again, two separate examiners in two separate provinces. Look at the essay. Growling sound of a dog, that was the growling animal behind, and is able to respond rapidly to the touch of its nose on his or her leg. Now, the second part is changed. Respond rapidly to the touch of the dog is behind this person and it touches this person. How does this person respond to that? So we're not going to look at the hearing part because we've already covered that in the last essay. So we're not going to waste our time with that. The second part is the reflex action. The receptor in the skin of the leg will receive the stimulus and convert it to an impulse which will travel along the sensory neuron to the spinal cord. What does it do there? It will go across the dorsal root of the spinal nerve. In the gray matter, the sensory neuron will make a synaptic contact with a connector neuron, which will make a synaptic contact with a motor neuron. What happens then? The impulses are then transferred via the motor neuron past the ventral root of the spinal nerve to the effector or the muscles in the leg, causing them to contract so that the person can run away. This is a reflex action. Sometimes because we are so chained with these things and you are associating that with that, you're not going to think about it, you're just going to turn and run. Now instead of saying pricking your finger, burning your finger, or tramping on a thorn, this examiner used the nose of the dog, the wetness of that, as a fear triggering mechanism that triggers a reflex action to run away from a source of fear. So the same thing happens there. It's a reflex action and they moved away there from there. Again, now let's look at the criteria. Again, let's look at this one. It says relevant to the topic. What was there? Only information relevant to hearing and reflex action. If you spoke about something else, you lost this mark. This one here, the process involving hearing, again, like the last one, they must move in sequence and in a reflex. You can't talk about a motor neuron, go back to the sensory and so on. And last but not least, at least half of both processes of hearing and reflex action are described correctly. Wow.
Wow, that's a lot of information, Aslam.
That's a whole lot of information.
Yeah, I hope you guys enjoyed the lesson. This does bring us to the end of the lesson today, but I really hope you guys have taken a lot from today's lesson. Remember, you can find all the material on our Facebook page. That's facebook.com/learnextra. On that page, you can find also entry instructions on how to win this Casio calculator. All you have to do is follow the link on the Test Your Knowledge page, enter the curio code, and you could be walking away with a brand new Casio calculator. Remember guys, also the water game, watergame.co.za, there's a chance there for the first place guy to win 5,000 Rand. So get over there and play that game. But until next time guys, have a good week and we'll see you later.